There is a moment that most people over 55 can pinpoint with uncomfortable precision. Not a dramatic moment, not a stroke, not a diagnosis, not anything that sends you to the emergency room. A quiet moment. You are mid-sentence, and the word you need, a word you have used 1,000 times, a word so ordinary it would embarrass you to forget it, simply is not there. You reach for it and find a gap. You pause, you talk around it, you say, "You know what I mean," and the person you are talking to nods and fills it in for you, and you laugh it off because that is what you do. You laugh it off and move on. But something does not move on. Something notices that it happened again. Maybe it is the name of the actor in the film you watched last week. Maybe it is where you put the keys you were holding 30 seconds ago. Maybe it is walking into a room and standing there with a specific, purposeful blankness, the intention that sent you there already gone. These are the moments that accumulate quietly through your 50s and into your 60s, and most people manage them with a combination of humor and private unease. And nobody tells them what is actually happening inside the 3 pounds of tissue behind their eyes. What is actually happening is this: your brain is losing volume. Not metaphorically, not in the sense of feeling foggy or distracted, physically, measurably. The human brain loses approximately 0.2 to 0.5% of its total volume every year after the age of 40, with the rate accelerating significantly after 60. By the time most people are 70, the brain is measurably smaller than it was at 50. The hippocampus, the region most critical for forming new memories and for spatial navigation, the region that Alzheimer's disease attacks first and most aggressively, shrinks by roughly 0.5% per year in people without active intervention. The prefrontal cortex, responsible for executive function, planning, and the retrieval of specific words during conversation, loses both volume and the white matter connections that allow it to communicate efficiently with the rest of the brain. This is not news to neuroscientists. They have known about age-related brain atrophy for decades. What has changed, what the research of the last 15 years has fundamentally altered, is the assumption that used to accompany this knowledge. The assumption that the shrinking was inevitable, that it was the passive consequence of biological time passing, like wrinkles or gray hair, something that could perhaps be slowed but not reversed, managed but not undone. That assumption is wrong. And the study that most definitively demonstrated it was wrong came not from a pharmaceutical lab developing a drug, but from a researcher at the University of Illinois named Kirk Erickson, who, in 2011, published a paper in the Proceedings of the National Academy of Sciences that genuinely changed how neuroscientists think about the aging brain. The paper was specifically about walking. About what happens to the hippocampus in older adults who walk regularly compared to those who do not. And what it found was not a slowing of atrophy. It was a reversal. The participants who engaged in aerobic walking for 1 year, 40 minutes, 3 times per week, showed a 2% increase in hippocampal volume. The control group, who did stretching and toning but no aerobic exercise, showed the expected 1.4% decrease. The difference between the two groups at the end of 1 year was 3.4% of hippocampal volume. The walkers did not just preserve their hippocampus. They grew it. Grew it! A structure inside the brain that neurology had long regarded as largely fixed in adult life, subject to progressive loss with aging, was demonstrably expanded by the simple act of walking briskly for 40 minutes, 3 times per week, for 12 months. The paper accumulated over 3,000 citations. It was covered by every major science publication. And then, in the way that findings which require behavioral change rather than pharmaceutical action tend to move through medicine, it was gradually absorbed into the background of what researchers know, and practitioners rarely have time to communicate in a way that changes what their patients actually do. This video is about what is happening inside the brain during that walk, not the outcome. The outcome is a 2% increase in hippocampal volume, and that is remarkable enough on its own. But the mechanism underneath the outcome, because understanding the mechanism is the difference between doing something because you have been told it is good for you and doing something because you understand exactly what it is doing to your brain in real time, which is the kind of understanding that actually changes behavior long term. The mechanism begins with a protein. A protein so central to the story of brain health and neuroplasticity that researchers who study it have started calling it miracle grow for the brain, which is an embarrassingly imprecise analogy that nevertheless captures something true about what it does. The protein is BDNF, brain-derived neurotrophic factor. BDNF is produced by neurons, brain cells, and by the surrounding support cells called astrocytes and microglia. It binds to receptors on the surface of neurons and triggers a cascade of intracellular events that promote neuronal survival, the growth of new dendritic branches, the projections that form synaptic connections between neurons, the formation of new synapses, and the process called neurogenesis, the generation of entirely new neurons from progenitor cells in the hippocampus. Adult neurogenesis, the idea that the adult brain can generate new neurons, was itself controversial until relatively recently. For most of the 20th century, the dogma was that you were born with all the neurons you were ever going to have, and the rest of life was a process of slowly losing them. That dogma was overturned quietly and definitively by a series of discoveries in the 1990s and 2000s demonstrating that the hippocampus continues to produce new neurons throughout adult life through a process driven primarily by BDNF signaling. BDNF is the signal that tells the hippocampus to grow, to produce new neurons, to form new synaptic connections, to maintain the dendritic density, the richness of the neural network that determines how quickly and accurately information can be retrieved from memory, how fluidly one thought can connect to another, how available language remains in the middle of a sentence. And BDNF levels decline with age. Significantly, measurably, beginning in the mid-40s and falling progressively thereafter. Low BDNF is associated with every neurodegenerative condition researchers have looked at: Alzheimer's disease, Parkinson's disease, depression, cognitive decline that precedes any formal diagnosis. The relationship is not incidental. Low BDNF means the hippocampus receives less signal to maintain itself. Less maintenance means more atrophy. More atrophy means the quiet moments in mid-sentence happen more often. Now here is the connection that makes the Kirk-Erikson walking study mechanistically coherent. Aerobic exercise is the single most potent stimulus for BDNF production in the brain that has been identified in human biology. Not supplements, not dietary compounds, not cognitive training, though all of these have genuine supporting roles. Aerobic exercise. Specifically, exercise that elevates the heart rate into the aerobic zone for a sustained period. The BDNF response to aerobic exercise is acute. Plasma BDNF rises during and immediately after exercise. And chronic, regular aerobic exercise produces sustained elevations in baseline BDNF that persist between exercise sessions, remodeling the hippocampus over months in the way Erikson's data demonstrated. The mechanism connecting exercise to BDNF has been worked out in substantial detail. Muscle contractions during exercise produce a compound called FNDC5, a membrane protein that is cleaved by the enzyme furin into a circulating form called irisin. Irisin crosses the blood-brain barrier and stimulates the expression of BDNF in the hippocampus. The exercising muscles are, in effect, sending a molecular signal to the brain telling it to grow. This is a remarkable piece of biology. The muscles in the brain are not separate systems operating independently. They are in continuous molecular conversation. A signal produced in the thigh muscle during a walk is crossing the blood-brain barrier and activating neurogenesis in the hippocampus. The body was designed, or evolved rather, over millions of years of obligate physical activity, with the assumption that the brain would receive this signal regularly. The brain's neuroplasticity mechanisms are calibrated to a level of physical activity that almost no modern adult is actually performing. The word-finding difficulty. The misplaced keys. The room you walked into with an intention that dissolved. These are not simply the unfortunate consequences of biological time. They are, in significant part, the consequence of a brain that has been receiving a signal it evolved to depend on at a fraction of the dose it was designed for. I want to introduce you to someone. Her name for this story, is Margaret. She is 67, a retired art teacher from the north of England, and she came to the attention of a neuroscientist at the University of Edinburgh, not as a patient but as a research volunteer. She had enrolled in a study examining cognitive function in healthy older adults because she had read about the study in a local paper and thought, in her words, that it was the kind of thing she ought to contribute to if she was going to spend her retirement complaining about memory research being underfunded. At her baseline cognitive assessment, Margaret performed in the 34th percentile for episodic memory, the kind of memory that allows you to recall specific events and experiences compared to age-matched norms. This is not dramatically poor performance. It is below average, meaningfully below average, but not the kind of result that would produce a clinical referral. It is the kind of result that sits in a normal aging trajectory and attracts no particular intervention. What attracted the researchers' attention was her BDNF. Her serum BDNF was in the lowest quartile for her age group, and her physical activity level, measured by accelerometer over 2 weeks, was characteristic of a sedentary lifestyle, approximately 4,200 steps per day on average, with no sustained aerobic activity. She was not particularly alarmed by this. She had not been sedentary deliberately. She had simply structured her days around the activities she enjoyed. Reading, painting, cooking, gardening, none of which elevated her heart rate into the aerobic zone for more than brief incidental periods. The researcher explained the BDNF exercise hippocampus connection to Margaret in the way researchers rarely get to explain mechanisms to research volunteers because they are normally trying to keep protocol conditions consistent. Margaret asked, with the directness of a woman who had spent 40 years managing classrooms full of teenagers, what specifically she needed to do and for how long. The answer was brisk walking. Enough to be slightly breathless, enough that you could still hold a conversation, but it would require a little effort. 40 minutes, 5 days per week. She found this unremarkable as a prescription and slightly annoying as a practical matter, because 40 minutes of brisk walking 5 days per week requires, as she put it, a level of organizational commitment that feels disproportionate to the activity. But she agreed to it. She was measured and tracked and assessed for 18 months. At 18 months, her episodic memory score was in the 61st percentile, 27 percentile points. Her serum BDNF had risen from the lowest quartile to the 58th percentile for her age group. An MRI of her hippocampus showed a volume increase of 1.8% over the baseline scan. She had gone from losing hippocampal volume at the expected rate to growing it. She described the cognitive change not as a dramatic transformation, but as a gradually accumulating clarity. The word finding improved first, within the first 2 months. The spatial memory, remembering where she had put things, navigating new places with less anxiety about getting lost, improved over the following 6 months. The most recent improvement, which she mentioned at the 18-month mark almost as an afterthought, was that she had started reading novels again. She had quietly stopped reading fiction several years ago because she found she was losing track of the plot threads and the character names, which made the activity frustrating rather than pleasurable. She had not connected this to her memory function. She had simply stopped reading fiction and shifted to non-fiction, which she found easier to follow because it did not require holding multiple simultaneous narrative threads in working memory. She had gone back to fiction. She was reading again. She mentioned the title of the novel she had just finished with the casual pleasure of someone for whom a lost enjoyment has been quietly restored, without fanfare, by something as undramatic as a daily walk. The conversation between the muscles and the brain through irisin and BDNF is real, but it is not the only conversation that exercise initiates in the aging brain. There are 4 others that compound the effect in ways that make the 2% hippocampal volume increase. Dramatic as it is, only part of what is being changed. The first is cerebral blood flow. Aerobic exercise increases cardiac output and blood pressure in a way that drives increased blood flow to the brain. This sounds unremarkable until you understand that the brain's blood supply is not uniform. Different regions receive proportionally different flow, and the hippocampus and prefrontal cortex, the regions most vulnerable to age-related atrophy, are highly sensitive to changes in cerebrovascular health. Chronically elevated aerobic exercise increases the density of capillaries in the hippocampus, a process called angiogenesis, meaning more blood vessels delivering more oxygen and more glucose to the most cognitively critical regions. A brain with higher capillary density in the hippocampus has greater metabolic reserve. It can sustain cognitive activity at higher intensity for longer before function degrades. This cerebrovascular benefit has a particularly important implication that the standard brain health conversation rarely addresses. It is one of the primary mechanisms through which aerobic exercise reduces the risk of vascular dementia, the second most common form of dementia after Alzheimer's, which is driven by cumulative small vessel disease reducing blood supply to critical brain regions. The daily walk is not just growing the hippocampus through BDNF. It is maintaining the vascular architecture that keeps the hippocampus and prefrontal cortex adequately supplied with the resources they need to remain functional. The second conversation is between exercise and the inflammatory environment of the brain. The brain, in chronic low-grade systemic inflammation, the state that characterizes most sedentary older adults with the metabolic profiles of modern life, is not operating in a neutral environment. Systemic inflammatory cytokines, particularly TNF-alpha and interleukin-6 produced by visceral adipose tissue and by the activated macrophages in artery walls, cross the blood-brain barrier and activate the brain's resident immune cells called microglia. Chronically activated microglia produce their own inflammatory mediators that impair synaptic function, reduce neuroplasticity, and suppress BDNF expression, creating a feedback loop in which systemic inflammation suppresses the very brain growth factor that would otherwise maintain hippocampal volume. Aerobic exercise is one of the most effective anti-inflammatory interventions known to human biology. The mechanisms are multiple: reduced visceral adiposity, improved insulin sensitivity, upregulation of anti-inflammatory interleukins, including IL-10 and IL-4, reduced NF-κB, signaling in peripheral immune cells. The net effect is a measurably reduced inflammatory burden on the brain, which allows microglia to return to their resting surveillance state and allows BDNF expression to recover from the suppression that chronic neuroinflammation produces. The third conversation involves the glymphatic system. The brain's waste clearance mechanism, discovered in 2013 by Maiken Nedergaard's group at the University of Rochester in what was one of the genuinely surprising neuroscience findings of the last 20 years. The brain has no lymphatic vessels in the traditional sense. Instead, it uses a system of channels that run alongside blood vessels through which cerebrospinal fluid flows during sleep, carrying metabolic waste products out of the brain tissue and into the peripheral lymphatic system for clearance. One of the primary waste products cleared by the glymphatic system is amyloid beta, the protein fragment that accumulates to form the amyloid plaques that define Alzheimer's disease pathology. Impaired glymphatic clearance means amyloid beta accumulates. Adequate glymphatic function means it is cleared nightly during deep sleep. The connection to exercise is through sleep architecture. Aerobic exercise is one of the most robustly demonstrated interventions for improving deep sleep, specifically the slow-wave sleep phases during which glymphatic clearance is most active. People who exercise regularly spend more time in slow-wave sleep, achieve deeper slow-wave sleep, and show higher rates of glymphatic clearance of amyloid beta during sleep than sedentary individuals. The daily walk is, through the mechanism of improved deep sleep, directly contributing to the nightly clearance of the protein that accumulates into the plaques of Alzheimer's disease. This is not a fringe hypothesis. The glymphatic amyloid connection has been replicated across multiple research groups and is now central to the scientific understanding of Alzheimer's pathogenesis. The exercise-sleep glymphatic axis is the mechanistic bridge between daily physical activity and reduced Alzheimer's risk, a relationship that has been observed epidemiologically for decades, but whose mechanism was not fully understood until the glymphatic system was characterized. The fourth conversation is the one that is perhaps most practically actionable because it has implications beyond just exercise. It involves the gut-brain axis, specifically the bidirectional communication between the gut microbiome and the central nervous system through the vagus nerve and through systemic circulation of metabolites. Aerobic exercise consistently produces favorable shifts in the gut microbiome, specifically increasing the relative abundance of lactobacillus and bifidobacterium strains, and of the butyrate-producing bacteria Faecalyobacterium prausnitzii and Roseburia intestinalis. These shifts are relevant to brain health through two mechanisms. First, butyrate produced in the gut reaches the brain and has direct neuroprotective effects. It inhibits histone deacetylases in neurons, maintaining the epigenetic landscape that supports BDNF expression and synaptic plasticity. Second, a gut microbiome dominated by beneficial bacteria produces lower levels of the gut-derived inflammatory metabolites, including lipopolysaccharide, that cross a compromised gut barrier and drive neuroinflammation. The exercise is improving the gut microbiome. The improved gut microbiome is reducing neuroinflammation. The reduced neuroinflammation is allowing BDNF expression to recover. The recovered BDNF is driving hippocampal neurogenesis. These are not parallel independent benefits of exercise. They are a cascade, each step enabling the next. Now, the specific details that determine whether the biology described above actually happens in your brain or whether you walk without producing the neurological effect. The heart rate zone matters. The BDNF response to aerobic exercise is dependent on exercise intensity reaching a threshold that corresponds to approximately 60 to 75% of maximum heart rate. Below this threshold, slow, gentle walking that does not elevate breathing, the irisin and BDNF response is significantly attenuated. The exercise needs to be brisk enough that a conversation requires slight effort. Not gasping, not so hard you cannot speak, just the edge of comfortable. In a person of 60, maximum heart rate is approximately 160 beats per minute, so the target zone is roughly 96 to 120 beats per minute. For most people, a genuinely brisk walking pace on a route with some mild incline achieves this without any particular effort calibration beyond paying attention to breathing. Duration matters. The BDNF response to a single aerobic exercise bout is linear up to approximately 40 minutes and then plateaus. The minimum duration that consistently produces measurable BDNF elevation in human studies is 20 minutes. The dose used in Erickson's study, like 40 minutes, sits at the optimal point of the dose-response curve. If 40 minutes 3 times per week is the realistic starting commitment, that is the starting commitment. The most important thing is that the commitment is started and maintained. A 40-minute walk 3 times per week is dramatically more effective than a theoretically optimal 40-minute walk 7 times per week that does not actually happen because the target was set above what the schedule realistically supports. The timing has nuance that most exercise recommendations omit. Morning exercise in a fasted state, before breakfast, produces a BDNF response that is amplified by the fasting-induced AMPK activation that is already present after an overnight fast. AMPK and the irisin BDNF pathway work through related but distinct mechanisms, and their combined activation in a fasted morning walk produces higher BDNF levels than the same exercise performed in a fed state. This does not mean the only useful walk is a fasted morning walk. Afternoon and evening walks produce the same structural changes over weeks and months. But if you are optimizing for acute BDNF response, the morning fasted walk is the conditions under which the acute effect is highest. Novelty amplifies the neurogenic effect of exercise. Walking the same route produces a BDNF and neurogenesis response. Walking a novel route, one requiring spatial navigation, attention to new landmarks, active engagement with an environment that has not been fully mapped, produces a measurably higher neurogenic response because hippocampal neurogenesis is not purely a metabolic event. It is also an experience-dependent event. The new neurons generated by BDNF signaling survive and integrate into the hippocampal circuit, preferentially when they are being actively used for spatial navigation and novel experience encoding. A walk through an unfamiliar neighborhood, a forest path taken in a new direction, any route that requires active attention to navigation. This walk is producing more lasting neurogenesis than the same walk taken on autopilot along a fully memorized route. This is perhaps the most practically useful refinement in the entire body of exercise and neuroplasticity research. Vary the route. Not every walk, not dramatically, but with enough regularity that the hippocampus is being challenged to encode new spatial information, which is both the most natural form of hippocampal activation and the activity for which it generates new neurons most efficiently. What Margaret does now, 18 months in, is walk for 40 minutes every morning before breakfast. She has mapped 15 different routes through her town and surrounding countryside. She does not repeat the same route within the same week. She has been doing this for long enough that some of the routes she established in the first months feel fully familiar, and she has started extending them into adjacent areas to maintain the novelty. She does this with the same practical commitment she brought to a 40-year teaching career, without sentimentality and without making it the centerpiece of her self-narrative. It is just what she does in the morning, like brushing her teeth, but with better outcomes for her hippocampus. She is reading her third novel of the month. The plot threads are holding. The character names are staying in place. The brain that is shrinking is not the brain that has to keep shrinking. The biology is clear on this. The mechanism is worked out. The clinical evidence is robust enough that it has been replicated across dozens of independent studies in multiple countries and has been specifically recognized by the National Institute on Aging in its guidelines on dementia prevention. The prescription, if it could be written on a pad and handed across a desk, would read aerobic walking, 40 minutes, 3 to 5 times per week, at a pace that makes conversation slightly effortful on varied routes, preferably before breakfast, for as many years as you have remaining. No drug does what this does to the hippocampus. No supplement produces the BDNF response that a genuinely brisk walk produces. No cognitive training app generates the angiogenesis, the neuroinflammation reduction, the glymphatic clearance optimization, and the gut-brain axis improvement that 40 minutes of elevated heart rate walking produces simultaneously in the same session through the same cascade of molecular events. The word that disappeared mid-sentence. The keys that were somewhere and are now nowhere. The room you walked into with a purpose that dissolved before you arrived. These are not inevitable. They are modifiable. The modification is free, requires no prescription, no equipment beyond a pair of shoes, and begins producing measurable biological effects in the hippocampus within the first week of consistent practice. The Harvard research called it remarkable. The Swedish Academy called the underlying neuroscience Nobel-worthy. Margaret calls it her morning walk. 40 minutes. Brisk enough to feel it. A route you have not taken before. Your hippocampus will do the rest.